I'm excited to be talking about some ongoing work to use multi-omics single-cell data to understand T-cell regulation in viral infection. So we all know that single-cell technologies have been really useful for understanding biology in new ways. We generally use single-cell RNA-seq to understand cell type heterogeneity as well as trajectories between different cell states. And we also use single-cell attack-seq for understanding the regulation of gene expression. And today I want to talk about how we can use both of these in a multi-omics single-cell attack and single-cell RNA-seq dataset of T cells responding to LCMV in a mouse model. And in particular, I want to present some new discoveries about T cells and that we use from analyzing a new single-cell attack-seq data and a new T cell regulatory atlas. So there's a lot of questions on how to best analyze single-cell attack-seq data that I obviously don't have time to go over all of these today, so I'm just going to focus on one of these, which is how do we determine the features that we want to use for quantifying single-cell attack-seq data. So we don't really think about this when we're analyzing single-cell RNA because we know exactly where our genes or features are, but for attack, we have to design a set of regulatory regions of interest. And so there are many ways you can do this, and people do this, and so it can get a little bit complicated. And so in this work, we tried to build a peak compendium using a bunch of different publishedly bulk attack data sets of T cells. And so we generated this new peak compendium, which can be used to analyze any future single-cell attack-seq data of T cells. We've also generated some epigenomic signatures for different T cell states, which allow us to extract new signal from our data set. So our data set is a multi-omic data set of splenic T cells responding to two strains of LCMV, one generating an acute infection and the other one generating a chronic infection. And using our bulk compendium, we can clearly differentiate between known T cell states and cleanly separate cells from different T cell lineages like CD4 and CD8. We can also use our epigenomic signatures, which I'm showing over here, to differentiate between similar T cells. Here I'm doing naive and memory. We also find that single-cell RNA-seq is actually worse at differentiating these different T cell states because it merges cells from our two different CD4 and CD8 lineages together due to transient effects like cell cycling. And so that's one little takeaway here is that actually single-cell RNA-seq can actually be better for identifying different cell states than single-cell RNA. In terms of more biological findings, there's been a lot of controversy in the field about how different the responses are to chronic and acute infection, and specifically pertaining to whether certain cell states exist in these two different responses. And here we can clearly see that the same T cell states exist in both the acute and the chronic infection. And I'd like to particularly highlight these two cell states, so the CD8 effector cells, which we see not only in acute infection as expected, but also in the chronic infection, and CD8 dysfunctional cells, which are very characteristic of the chronic infection, also exists in the acute infection. And we can, again, use our epigenomic signatures that we created to show that the cells that I have labeled as CD8 dysfunction are indeed enriched for this dysfunctional score in both the acute and the chronic response. We also use hierarchical clustering to show similarities between different T cell states based off of both peak accessibility profiles and gene expression patterns. And we can see that, surprisingly, T cells from the different lineages can often be more similar to each other than they are to T cells from their own lineage. So I want to highlight an example of this, which is the CD4 T follicular helper cell population, which is more similar to CD8 progenitors than to any other cell state. And so we're very interested in this finding because we think these cell types are very interesting. So these TFH cells are known to interact with B cells. In fact, many of their marker genes were originally discovered in B cells. And the CD8 progenitors are this clinically relevant stem cell-like population that are known to respond to immunotherapies and improve cancer patient survival. And so we've used archetypal analysis as a way to show that these share a sort of regulatory program. And we also hypothesized, based off of the fact that our progenitors are also expressing B cell genes, that perhaps they're sharing some sort of regulatory program, perhaps through some sort of direct interaction with B cells or some shared microenvironment in each. So I hope this is kind of giving you a sense of how we can use multi-omic data, and especially single-cell attack-seq, to understand the heterogeneity of T cell states and uncover regulatory programs shared between them. And with that, I'd like to thank my lab and my advisor, and especially our collaborator, Joris van der Viken, who generated all of this data. And the lab is hiring, so talk to Yuri if you're interested.